Uh, Good morning. Just confirm me, Basha, am I audible to you? Okay, fine. We'll start at today's class. See, in this class, yesterday we have started developmental biology and we have complete two processes, spermatogenesis and oogenesis. In today's class, we are going to discuss about fertilization and cleavage, gastrulation process, and parthenogenesis process, right? Okay. See, what is fertilization? A fusion of male gamete and a female gamete, it is called as a fertilization. As a result of this particular fusion of male and female gamete, we are going to get a structure called a zygote. Zygote is a um, fused cell of male and female gamete. Okay, that is what this process is called as the fertilization. And where this fertilization is going to happen? In the female reproductive part, most specifically in the fallopian tube. Okay, and what are all the things which is happening in the process of fertilization? So the sperm has to be, if a fertilization has to be happen in the fallopian tube of female reproductive system, first thing it has to be reach the egg. Okay, to reach the egg, the sperm has to enter into the female reproductive part and it has to penetrate the outer layer of the egg, which is called as the zona pellucida. This must be your questionable area. Okay, so this is the first area where the egg is going to break the layer and enter, where the sperm is going to penetrate into the egg by breaking this particular layer. Now, after the penetration of this through this particular layer, the sperm and the egg membranes are getting fused. Okay, the cell membrane of sperm and the membrane of the egg is getting fused. Now, as a result of this fusion, we are going to get a diploid zygote with a complete number of chromosomes. We know that sperm is haploid in nature, ovum is also haploid in nature. As a result of this fusion, we are going to get 2n, and this 2n is said to be as the zygote, which is in diploid condition. Now, how long this process of fertilization is going to take? Now, this generally occurs shortly after the ovulation process within 12 to 24 hours of the release of the egg. Okay, so it happens within 12 to 24 hours. Now, what are all the things which affecting the fertilization? Sperm viability and motility. See, if the sperm doesn't, if the sperm is sterile, and then the fertilization is getting affected. Now, the timing of ovulation and sperm availability. See, the egg has been released shortly after the ovulation. Okay, and if the fusion occurs after some time, and it is depends on the availability of sperm, it depends on the count of the sperm and also at the time of ovulation. Okay, and based on this phenomena, the fertilization is taking place. Now, health and receptivity of the female reproductive tract. Okay, if the sperm enter into the female reproductive tract, the egg has to be ready to receive the sperm. Okay, so if that is getting affected, then the fertilization is also not takes place. Okay, now what are all the importance of this fertilization? It leads to pregnancy, right? Now it ensures a genetic diversity through the recombination. So we know that male are growing in a different area, different locality, and female is also in a different, completely in a different area apart from the consanguineous mating. They are going to fuse their sperm and the egg, right? And this leads to some of the, it leads to a mitotic division and meiotic division. In the meiotic division, we have studied about the crossing over, and this crossing over leads to exchange of chromosomal material between these two cells. And as a result of this process, we are going to have a variety of genes, genetic diversity. Okay, now what will be the end product? Formation of a zygote. So you should know that fusion of male and female gamete will be the process of fertilization, right? Now, and as a result of this process, what will be the end product? As a result of this fertilization, we are going to have a zygote. Okay, and after that, this zygote undergo a cell division, and that cell division is called as a cleavage. And after the cleavage, it leads to a stage of blastula. And after the stage of blastula, it leads to a gastrula, which means what? A formation of three germinal layers. And after that, it will be implanted into the uterine wall. Okay. And what are all the assisted reproductive technologies? So if the normal fertilization is not happening, then there will be an assisted method which is also there, like in vitro fertilization, where the fertilization occurs outside the body. Okay, it will be fertilization will be happen in the laboratory. And what will be the measures to avoid the pregnancy? Contraceptive methods such as a barrier method using the condoms, and hormonal methods, that is birth control pills, intrauterine device IUDs. Okay, so these are all the contraceptive methods. It is a birth control process, right? Now, what is meant by cleavage? We have studied about the fertilization and we know what is zygote. Now, what is happening after the formation of zygote? See, you should know that cleavage is nothing but a cell division. And what is the cell division is taking place? Whether it will be the mitotic cell division or meiotic cell division. That will be your questionable area here. That will be a rapid mitotic cell division of the zygote. Okay. Now, after the fertilization without overall growth of the embryo. See, fertilization fusion has been completed, and as a result of fusion, zygote has been formed. And after the zygote is getting formed, it results in the rapid cell mitotic cell divisions. Okay. Now, what is the purpose of having this cell division? Increase in number of cells while maintaining a constant total volume of the embryo, preparing for the formation of a blastocyst. Okay, now what are all the types of cleavage? We have a different types of cleavage. One is holoblastic and meroblastic cleavage. Not only this cleavage, we have a lot of cleavage. And see, this holoblastic cleavage, it is occurs in egg with little or moderate amounts of yolk, resulting complete division of egg into the smaller cells. That is what a holoblastic cleavage. The word holo means complete. Okay, complete cleavage. The entire zygote undergo a division and it results in the formation of smaller cells. Meroblastic cleavage. Here in holoblastic, we have a little or moderate amount of yolks. In meroblastic, we are having a large amount of yolk. Here, the cell division is incomplete due to the presence of this larger amount of yolk. Okay, now some of the examples for this holoblastic cleavages: mammals, including the humans, they are undergoing a holoblastic cleavage. As a result of this cleavage, we are producing a solid ball. This solid ball is said to be as the morula stage. Okay, and also in amphibians, this also cleavage leads to the formation of blastula stage. In the meroblastic cleavage, it is happening in the birds and the fish. In birds, the initial cleavage occurs only in the yolk-free region. Okay, so that cavity is said to be as the blastodisc, forming a blastoderm. Okay, now in the fish, this cleavage occurs around the yolk mass, forming the blastodisc. Okay, so you should know holoblastic cleavage is taking place in mammals and amphibians. Meroblastic cleavage is taking place in birds and the fish. Right now, what is meant by gastrulation? Here, gastrulation means formation of three germinal layers, and that is ectoderm, mesoderm, and endoderm. Ectoderm is an outer layer, mesoderm is a middle layer, endoderm is an internal layer. Okay, this is what the formation of three germinal layers is said to be as the process of gastrulation. Now, typically occurs after cleavage and before organogenesis. It triggered by signaling molecules and positional cues. Okay, so this after, see, initially fertilization, and after that zygote is getting formed, and after the formation of zygote, cleavage, the rapid mitotic cell division is taking place, and as a result of this cleavage, we are going to get a blastula, and after that gastrula, the formation of blastula. This process is said to be as a blastulation, and formation of gastrula is said to be as a gastrulation. And after that, formation of an organs, that is said to be as the organogenesis. And this gastrulation is triggered by a signaling molecules and positional cues. We know that, and we are going to study about that. And what are all the key events taking place in the process of gastrulation. First thing will be the invagination. In invagination is nothing but it is growing inward. This, okay, like this is what an invagination. The cells at the blastula surface move inward to form a primitive gut, that is archentron. All right. Now what is germinal layers? Here the differentiation of cells into three germinal layers, that is an outer layer. So during the invagination, some of the cells grows as an ectoderm, some will be grown as an endoderm, and some will be present in between these two layers will be the mesoderm. That is what a formation of germinal layers. This particular process is said to be as the gastrulation process. So you have to focus here that ectoderm gives rise to what are all the area. This ectoderm gives rise to skin and nervous system and other related to these structures. And mesoderm will be forming the muscles, skeleton, circulatory system, and other internal organs are arising from this middle layer. That is mesoderm. And from the endoderm, it is an innermost layer. It is develops into an epithelial lining of the digestive tract, respiratory tract, and associated organs like liver and the pancreas. So you should know what are all the organs are arising from which layer. Okay. Why this gastrulation has to be happen? Because a basic body plan and sets the stage of the organ formation and subsequent development. This is what a basic process for cell to differentiate and formation of tissue and formation of organs. Now what are all the types? Invagination, involution, ingression, delamination. So you should know all these technical terms and you should know the meaning of all these terminologies. So what is invagination? We know that inward movement of the cells. Okay. So involution. Here, the cell migrates inward and spread breadth out the outer layers. It is what a rapid inward movement. Okay. That is what an involution. And this migration towards the inward by the cells, it is spread beneath the outer layers. Okay. There will be a lot of layers which will be involute under the outer layer in the insects and the mollusk. And what is ingression? Here, the cells migrate into the embryo from the surface. From the surface is getting migrated. That is what an ingression. Delamination. See here, the splitting of one cellular sheet into two sheets. See, for all these terminologies, whenever you have listened to the class of Sir's class, developmental biology, I hope he showed the diagram. And you have to go through the picture and you have to remember accordingly. Okay, only then you can't forget. Okay, so all these words you have to go through the picture, okay, which is there in the PPT, right? Now, so you should know imagination is seen in the amphibians and echinoderms. Involution is seen in the insect and mollusk. Ingression, vertebrates. Delamination in birds and the mammals, that is humans. Okay. Now, what is parthenogenesis? See, up to now we have studied about a male and female gamete is getting fused. As a result, we are going to get a zygote. This is what this fusion is said to be as a fertilization. Now, here in parthenogenesis, there is no process of fertilization is going to take place. So, instead of that, it is a form of an asexual reproduction. Here, a egg is not getting fertilized, which means what? It is not meeting any of the male gamete. Now, this unfertilized egg, it is developed into a new individual without the process of fertilization by the male gamete. They themselves develop into a next, they have the capacity to produce a next generation. Okay, and they have their own pros and cons, that is different. But here this egg is not getting fertilized and the unfertilized egg has a capacity to produce a next generation. Now there will be a different types of parthenogenesis, and first will be the artificial, natural, automixes and apomixes. So all these terminologies are very, very important, and we can find all these terminologies frequently in the question paper. So what is for the artificial parthenogenesis? Mean that will be induced in the laboratory through certain chemicals or electrical stimulation of an egg. Okay, that is what an artificial that will be done by the humans. A natural parthenogenesis. It is not under the stimulation of any external factor. It occurs naturally. It occurs spontaneously under various environmental conditions. Okay. Now, automixes. Here, this parthenogenesis involving meiosis, and we know that in meiosis, crossing over is taking place. Then obviously, a recombination is also taking place. That is what an automix is. This type of parthenogenesis seen in reptiles and amphibians. Now, what is apomixis? Here, parthenogenesis is occur without meiosis and recombination, and that will be happen in the plants and invertebrates. So these are all the types of parthenogenesis. Right now, how this process is going to take place? Here, the activation of a cell's developmental program without fusion of sperm typically involves the activation of X cell receptors or chemical signals. Okay, so the egg themselves are getting activated by some of the developmental program. Okay, by activating their receptor through the chemical signals. Okay, only by this process they can have the capacity to give rise to a next generation. Now, where we can find all these processes? Insects, crustaceans, fish, reptiles, amphibians, and certain plants. Okay, so these are all the examples we can find the process of parthenogenesis. So what are all the pros and cons? That is advantage and disadvantage. In advantage, that will be the efficient reproduction without a need of a mate. See, for if the process of natural fertilization has to happen, definitely they need a male and they need a female. Then both the egg and male gamete has to fuse. But this process, there is no need of any other partner. Okay, the female has the capacity to give rise to the next generation. So that is, they don't want to depend upon the male to get fertilized. Now here, they allows rapid colonization of new environments. Okay, but this preserves favorable gene combination in isolated population. So these are all the advantage. Now what are all the disadvantage here? Wait a minute. And here in this disadvantage, they have a limited genetic variety, genetic diversity, because they didn't have a male. They didn't find a fusion with male, and they see if it is not getting fertilized, what are all the characters present in the mother? Okay, only that character will be passed to the next generation. We don't find any of the advantage will be acquired by that particular individual. If it is getting fused with the male, obviously that results in the newer character. But in this parthenogenesis, we have a little genetic diversity. At least we have no genetic diversity at all. Now, it leads to a vulnerability to the environmental changes and diseases due to lack of this genetic variability. Okay, and also there will be a deleterious mutations over the generation. Okay, so these are all the disadvantages. So answer for this question: which of the following describes fertilization? That will be the fusion of an egg and the sperm cell. Where does fertilization typically occurs in humans? That will be in the fallopian tube. Where does fertilization usually occurs relative to ovulation? That will be after ovulation. Okay, within 12 to 24 hours after the ovulation. Okay, you have to keep in the number timing also. That is important, because only at that particular time the egg is viable. Okay, when at that particular time when it meets the sperm, the fertilization is easily going to happen, because at that time the egg is very effective and efficient. Which of the following is a crucial factor after affecting the successful fertilization? Number of eggs released, size of the uterus, health of a fallopian tube, blood type compatibility. That will be health of the fallopian tube. What is the end result of fertilization? That will be, see, here as a result of fertilization, we are going to get a zygote. Zygote is undergoing a cleavage. As a result of cleavage, we are going to have a blastula, that is blastocyst, gastrula, everything. Okay, at this particular time, we are going to form the blastocyst, and the blastocyst has to implant in the uterine wall, that is endometrium, and there will be the development of placenta also. So all these will be only happen and the post fertilization. In the post fertilization, all the phenomena is going to take place. Only that, see, for all these things to happen, the only process has to take place is the process of fertilization, right? So be mindful when I answer the question. Which reproductive technology involves fertilization occurring outside the body? That will be in vitro fertilization. What prevents fertilization from occurring naturally during certain times of the menstrual cycle? What prevents the fertilization from occurring naturally during the time of menstrual cycle? For that, you should know what are all the hormones involving, and which hormone involving thickening the endometrium. So all these things are very important. And that will be a presence of cervical mucus. See, the cervical mucus changes throughout the menstrual cycle, becoming more hospitable to the sperm around the ovulation and less so at other times, which affect the fertilization. Okay. Which component of the sperm cell is primarily responsible for penetrating the egg during the fertilization? Sperm has a DNA or not? Do the sperm have a DNA or what? And where the DNA is present, in which part of the sperm? See, we can divide the structure of the sperm as head, middle piece, tail. Okay, and that will be in the head. Okay, so the answer for this question is acrosome. And what will be the name of an enzyme which is present inside the acrosome? And that will be hyaluronidase enzyme, right? Which part of the egg does fertilization typically occur? and that will be zona pellucida. Okay, we know that this is the layer where the sperm is penetrating through this particular layer. Okay, what is the term for the event where the genetic material from the sperm and the egg combine to form a zygote? This is nothing but a fusion, that's all. That is syngamy. See, it refers to the fusion of sperm and egg nuclei which occurs during the fertilization, during the formation of zygote. Very good. Now, what is cleavage in embryology? See, we have a lot of questions to discuss. Kindly, please, fast, little bit. We have a lot of questions to discuss. Okay, and that will be rapid cell divisions of the zygote. Which of the following describes a holoblastic cleavage? Here, that will be the incomplete division due to the large yolk mass, division only in the region without yolk, complete division of the entire egg into smaller cells, formation of blastula with yolk sacs. And that will be obviously a complete division, and that complete division results in the formation of smaller cells, that is micromeres, in which organism the meroblastic cleavage typically occurs. So examples very important. Okay, give me the answer. And that will be birds. Okay, this meroblastic cleavage that will be occur in the birds where we can find a larger amount of yolk. Okay, and the cleavage where only the yolk-free region, this yolk-free region is the one undergoes a cleavage, that is blastodisc. Clear? What is the purpose of cleavage during embryonic development? Formation of placenta, differentiation into germ cells, increase in number of cells, formation of blastocyst. That will be to increase in number of cells. Okay. Which type of cleavage involves division around the yolk mass? That will be a meroblastic cleavage. Just go through the slides. During cleavage, what is the term for a solid ball of cells formed in mammals? No doubt in that. Obviously, it will be morula. Which type of cleavage is characteristics of insect like Drosophila? Very good. That will be the syncytial cleavage. Okay, see in insects like Drosophila, the early cleavage involves a rapid divisions without cytokinesis. And where we can find the superficial cleavage is seen in, of course, it will be the insects. Okay, so don't get confused with the superficial and syncytial cleavage. Okay, now, what process follows cleavage during embryonic development? We have 10 minutes more. That will be gastrulation, formation of three germinal layers. Which type of cleavage involves cells migrating inward and spreading beneath the outer layers? These are all the straightforward questions. You can't avoid these. That will be involution. Okay, it migrate inward and spread beneath the outer layers, which is seen in the insects and mollusks, right? In which organism does radial cleavage typically occur? That will be echinoderm. So you should know that radial cleavage, it is a characteristic phenomena of the echinoderms. Uh, you know what is gastrulation? Is formation of three germinal layers. Okay, which embryonic process follows a gastrulation? Okay, so this is what an order, right? Fertilization, zygote, followed by cleavage, followed by blastula, gastrula, and organogenesis. You should not forget all these things. Okay, which during gastrulation, what structure forms the cells migrate inward to create a primitive gut? And that will be orchentron, right? Which germ layer gives rise to the nervous system and skin epithelium? That will be obviously the ectoderm. Okay, so you should know which layer gives rise to which organs and all. During which stage of embryonic development does gastrulation typically occur? At which stage? At blastula stage. Okay, that is the one followed by gastrula. Okay, answer this question. Which process involves the differentiation of cells into specific types to form tissues and organs during gastrulation? We have 4 more minutes. Kindly make it fast. That will be organogenesis, no doubt in that. And which layer gives rise to muscles, bones, and circulatory system? That will be, no doubt, that will be mesoderm. Okay. Which type of gastrulation involves cells moving through the blastophore to form a germ layers? and which process? Just read the definition of ingression, invagination, involution, okay, delamination. So that is very, 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 very important. Here the cell is getting moving through the blastophore to form the germ layers, that is called as an ingression. Okay, kindly go through the definition. What is parthenogenesis? That will be the asexual reproduction without fertilization by a male gamete. Which type of parthenogenesis involves meiosis and recombination of genetic material? That will be automixis, right? In which organisms is parthenogenesis commonly observed? In all the things, in all the organisms, we can find the parthenogenesis very often. Okay. And what will be the advantage of parthenogenesis? That will be rapid colonialization of the newer environments. Okay, that will be the biggest advantage in that. And which reproductive technology involves the artificial induction of parthenogenesis in eggs? Which technique? What will be the name of technique? And that will be artificial parthenogenesis. Okay, don't think much. It is a basic level question, right? Okay, fine. That's all for today. And for tomorrow's class, you have to study and come the embryogenesis in vertebrates. Okay, any doubts? Any clarification you want to know? Everybody? Okay, then let wind up the class. If you have any doubts, you can post it in the group. Thank you.